scientists used AI to create 16 new viruses. Not simulations, not models on a screen, 16 real physical viruses that had never existed on Earth before, written by a machine and then built in a lab where they worked exactly as designed. And I want to be careful with that sentence because the headlines around this story have been doing a lot of heavy lifting. These viruses cannot infect you. They cannot infect any animal or plant. But what happened in that lab is still one of the genuine turning points in modern biology, and the researchers who did it said so themselves. So today we're going to walk through what they actually built, how they built it, why some scientists are calling it a milestone and others are calling it a warning, and what it means for the next few years of medicine. If you appreciate this type of content, hit that like button. It genuinely helps this channel reach more curious people. And let's dive into it right now. So here's what happened. A team at Stanford University and the Arc Institute, led by a researcher named Brian He, asked an AI system to write viral genomes from scratch. The AI produced hundreds of candidate designs. The team narrowed those down, chemically synthesized the DNA for 302 of them, and introduced that DNA into E. coli bacteria to see if anything would happen. 16 of them came alive. They assembled themselves, they replicated, and they killed their bacterial hosts, which is exactly what a working virus is supposed to do. Now I know what you're thinking, because it was my first reaction too. This sounds like the opening scene of a disaster movie. But the specific type of virus matters enormously here. These are bacteriophages, and a bacteriophage is a virus that only attacks bacteria. It has no mechanism for entering a human cell. Phages are in fact the most common biological entity on the planet. There are trillions of them on your skin right now, and they've been there your entire life. So how does an AI write a genome? The team used two models called EVO1 and EVO2, and the easiest way to understand them is that they are ChatGPT for DNA. A language model reads enormous amounts of text and learns which word tends to follow which. These genome models read enormous amounts of genetic code, roughly 2.7 million bacterial and phage genomes on the order of 300 billion DNA letters, and learned which chemical letter tends to follow which. And that's the part I find fascinating. Nobody sat down and told the AI the rules of virus construction, because we don't fully know those rules. The model inferred them, the same way it would infer grammar. They pointed it at a virus called Phage 174. That choice was deliberate. Phage 174 is one of the simplest viruses known to science, 11 genes, about 5 in 400 base pairs. It's also the most studied virus in history. It was the first DNA genome ever fully sequenced back in the 1970s, which meant that if the AI invented something new, the team would immediately be able to tell how new it was. If this is the kind of story you want more of, drop a comment below telling me which part surprised you most. I read every comment, and the most requested questions usually turn into their own video. Here's where it gets genuinely strange. Some of these AI-written viruses didn't just work, they worked better than the real thing. A cocktail of the AI-designed phages was able to rapidly kill E. coli that had already evolved resistance to natural phages. And when the team put one of the designs under a cryo-electron microscope, they found it had built its protective shell using a component borrowed from a distant relative, a combination evolution had never produced on its own. A few of the designs were different enough from anything cataloged that they'd arguably qualify as new species. That's the headline underneath the headline. This wasn't a copy machine. It produced solutions that 4 billion years of natural selection had not. And that matters medically because antibiotic resistance is killing people right now. Phage therapy, using viruses to hunt bacteria that drugs can no longer touch, has been around for a century, and its biggest weakness has always been that bacteria adapt to a single phage very quickly. As Brian He put it, once that happens, it's game over for the medication. A cocktail of genetically distinct phages is much harder to escape. And if you're fascinated by the mysteries of history, archaeology, and the science behind the world's greatest discoveries, subscribe to ConsciScience and join thousands of curious minds uncovering the truth, one mystery at a time. This channel exists to take stories exactly like this one apart carefully, without the panic and without the hype. Now let's talk about the part that has security researchers uneasy, because it deserves an honest look. The team did build in safeguards. They deliberately excluded viruses that infect humans and other complex organisms from the training data, and the phage and bacterial strains they used are non-pathogenic with a long history of safe laboratory use. That's a real precaution, not a press release. But there are two open problems. The first is that anyone with enough computing power could, in principle, repeat this process without those exclusions. The second is subtler and, honestly, more interesting. DNA synthesis companies screen orders against databases of known dangerous sequences, and an AI-written genome can look so unlike anything cataloged that the screening software reads it as ordinary. Researchers at the Johns Hopkins Center for Health Security put it plainly in a companion piece in Science. The capability to compose viral genomes with generative AI now exists, and the governance to steer it safely does not. So where does this leave us? As of right now, the verified count is 16 working AI-designed phages, all targeting E. coli, all confined to laboratory dishes, none used in any patient. The genomes these systems can write are still tiny compared to a bacterium, let alone anything that infects a human. One of the researchers described the next step as AI-generated life, and he wasn't being dramatic. A genome is the instruction set for an organism, and a machine just wrote a working one. The real decision now sits with regulators and DNA synthesis companies, not with the lab bench. So tell me, what are your thoughts on this groundbreaking discovery? What are the implications for mankind? Thanks for watching, and I'll see you in the next one.